Medicine and Hygiene. Is it London School? Is it LSU HSC in New Orleans, Los Angeles? And he's going to talk about the novel target in oncogene-driven tumors. The biodata of Professor Koul is right on the screen. He's the head of GU Oncology Research and Associate Director at LSU LCMC Cancer Center in New Orleans.
So, I mean, this is a tough act to follow. How do you speak after Dr. Talwar gave you a life's worth of information and people still have questions about how do live bacteria cause changes in the mouse? I'm speechless whether I can even talk after this, but it's a tough act to follow. I would like to move your attention back to the basics. We will talk about EIF4G1 as a new potential target for oncogene-driven tumors. So I have no commercial disclosures. In the basics of protein, the central dogma of the protein synthesis, you have a genomic signal in the DNA which gets translated into the proteins, and the proteins perform the function. We have translational control, transcriptional control. If you have been in this meeting for the last couple of days, you have seen how gene expression change, thousands of genes change, but all the transcribed messages are not translated into protein. There is regular control of that process. So oncogene addiction in cancer. So I was hearing my own echo here, sorry about that. So oncogene addiction in cancer is well recognized. There are in tumors, you have multiple gain of function or loss of function mutations as a result of which are considered to contribute to tumorigenicity. Yet, a single activated oncogenic pathway or an oncogenic protein can drive the tumors in a majority of the tumor cases, and all these malignant properties are driven by just one single oncogene. Case in point is mutated RAS in pancreatic cancers. You have P53 mutations which can drive the whole process. But this concept of oncogene dependency has been invigorated and has been tested in many genetic mouse models as well as in many of the xenograft models where the cells have been mutated, and this is well accepted. But most of these pathways remain unregulated. There are multiple of these genes that are changed, and having the drug targets for them has become a difficulty. So transcriptional regulation has been studied at multiple levels. We have a transcriptional regulation, but the translational control of oncogenic process remains still to be understood very well. So what is translation? It is taking the simple messages that cell makes and converting it into the proteins. So you know that one of the proteins that is well studied in this regard is EIF4E, which is a target of mTOR signaling, and EIF4E is bound to 4EBP, which keeps it in the cytosol and does not let it to translate messages. But then once 4EBP gets phosphorylated, this complex gets formed, which brings in together the central component of this translational complex is EIF4G, which is nothing but a scaffold protein which brings in EIF4E message along closer to the 4A, which helps to bring this whole complex together, and then poly-binding protein forms the whole complex. Now one would say, why is this needed even at all? So why is the cap and tail in messenger RNA very important? Because it is required for regulating nuclear export of the messages. It helps in prevention of degradation of exonucleases and also promotes translation in multiple other ways. The poly-A tail is required to prevent RNA from enzymatic degradation. It also aids in transcription and termination and export of mRNA from the nucleus. Almost all eukaryotic mRNAs are called mRNAs because they have a poly-A tail. You use poly-CTT to isolate them. Half-life of mRNA has been calculated by multiple methods, and if a message is without a cap and a tail, it has about half-life of 31 minutes, and without a cap but the poly-A tail, half-life is 44 minutes. Without a tail but with a cap only, it is 53 minutes, but if both cap and tail are present, it is about 100 minutes. It doesn't make much difference 30 minutes or 100 minutes, but if you look at how many protein moieties or how many polypeptides are made out of a single message, from 2900 to 1.3 million. So this is very, very crucial because oncogenes are required in very high quantity. So if you have ability to interfere this process, especially if you look in RBCs, these messages can last as long as 120 days by this process. So cancer cells have taken advantage and have hijacked this process to have their high level of addictive oncogenes for themselves. So this is a structure kind of the mouse is difficult to handle here, but I will try still. So EIF4G is the scaffold protein which brings in poly-binding protein on one side and 4E on the other side with this message, and then ribosomes get attached to it, so it helps load the messages to ribosomes. There is also a cap independent part of the translational machinery, which is an iris mediated, where everything is same except that you don't need 4E to bring messages to the ribosomes. In terms of the translational control from the signaling pathways, I alluded to it before that this 4EBP, which is bound to 4E, keeps this message from coming into the complex. And when the mTOR is active, it phosphorylates this 4BP, and this 4EBP falls off, so you get 4E message to this complex and translation takes place. Yet, many of the inhibitors of the TOR pathway have not proved to be effective in preventing translation, suggesting that there is this phosphorylation independent way of activating transcriptional regulation here. So there are other 4G as 3 isoforms, G1, G2, and G3. G1 is predominantly overexpressed in the tumors. G2 plays part in non-cap dependent translation, and G3 is present in multiple other tissues and plays part in cap dependent translation. So why target then translational control? So as I told you that most of these pathways that are crucial for cancer and developmental progression require translational control, and specifically targeting translation of specific proteins. How does the cell decide? Cancer cell decide I'm going to only translate these few messages and leave others out. So the selection bias is again handled by this translational control. So we looked at one of the proteins in this, we looked at all the proteins in the translational machinery, and we observed that EIF4G1 was the only protein which is selectively increased in prostate cancer, and especially during prostate cancer progression. There's a protein level is very high. You can look in clinical cohorts. We looked at TCGA data and other metastatic prostate cancer data, the prostate cancer, neuroendocrine prostate cancer. In each case, we found both amplification and overexpression of this EIF4G1. And also, we found that patients who had high EIF4G1 had a poor survival as compared to the other patients. Then we thought, well, that is enough. Let's look at all other tumor cells. So we looked at multiple tumor biopsies and tumor tissues in the tumor microarray, and we find almost every solid tumor has upregulation or increased expression of EIF4G1. And this is the message level, again, unbiased databases which have been generated by millions of dollars by public funding. If you scan through them, you can find in almost every solid tumor there is multiple fold upregulation of the EIF4G1 protein. It is not only present here, but you look at survival across all these tumors, high levels of this EIF4G1 protein are associated with poor survival. In each case, not a single case that we could find in multiple tumor types. We also looked at other ways of looking it through the cell lines and each tumors, why this is up, is it the message is up, or there is a fusion of the genes, and in all cases there are multiple mechanisms that result in upregulation of EIF4G1. We looked at all cancer cell line data and find that all cell lines that are in the cancer genome database, we find there's an upregulation of EIF4G1. We also looked at the protein work, and we published this work a few years back. In 2019 we published, and we, based on these data, we hypothesized that EIF4G1 is potential target for therapeutic intervention. 2 years later, there was another paper from Gerhard Wagner's paper, which did the deep computational analysis with all the powders they had at Harvard, and they came up with exactly the same conclusion. Of all the proteins that you could look, the EIF4G1 is the one of the strongest candidates for therapeutic intervention. So how we study it in the lab, going to the basics, we look at effects in protein translation. You look at how much message is on polysomes versus monosomes, and then you try to interpret these data by looking at either single messages by PCR, or you can do the sequencing of these, which is called polysome profiling, and then you isolate these polysomes from monosomes by using differential centrifugation, and techniques are well established, nothing new there. So when we knocked down EIF4G1, we found that although the polysome loading was impaired in many cases, but there were still some messages that were being translated. But in each case, shown here is data from 2 different cell lines, that the loading of messages in these tumor cells was reduced when EIF4G1 was inhibited. So was the characteristic of cell growth, colony formation, cell migration, and invasion. Features that we look at were they were affected by or impacted by EIF4G1 knockdown. 2 of the proteins that we looked at that time point were N-cadherin and SNEL. Both are associated with transition to the mesenchymal cells or mesenchymal behavior. We found that those proteins were also decreased in their expression. We hypothesized that in normal cells, EIF4G1 drives this unique pathway of cap-dependent translation, but as the tumor progresses, because EIF4G levels are highly upregulated, they drive these processes of proliferation, clonogenicity, cell cycle progression, EMT and stemness, and therefore maybe promoting cap-dependent translation, which causes therapy resistance. Well, it is not as simple as that. It was a simple bottle. This is from Trends in Cancer. People, when they look at the literature, they find that there are more than this cap-dependent translation can be affected because the protein synthesis is inhibited due to energy supply, and EIF complexes may not be stable under those conditions. There are the pathways that have been used and the drugs that have been used to target metformin, platinum, Inc-128, rapamycin, and starvation simply to affect the mTOR complex, yet the ternary complexes are still formed and you still get translation of the proteins. In terms of summarizing this Trends in Cancer paper, they said that energy supply decreases as the tumors go from normal cell to the carcinoma and CETO to the highly invasive tumors, and this is because there's a plasticity of the cells is increased, and there's a translation of reprogramming from this normal cap-dependent translation to only stress-accepting factors being selectively adapted. So we went to test again. We took the cell lines which were therapy resistant, which is an enzalutamide resistant prostate cancer, and wanted to test whether EIF4G1 still played role under these conditions. We find that indeed elevated levels of EIF4G1 are present in these enzalutamide resistant cells, and if you block it, you again can inhibit their clonogenic activity as well as their proliferation rate and migration and invasion. And a small molecule which Wagner had developed, EI4EG1, again it is not really a specific and selective molecule as a dirty inhibitor, but it does work. We observed that it was not really inhibitor of EI4G1, but it was actually decreasing the levels of this protein of EI4G1 itself. And so this EI4G1 decrease was sufficient to limit the metastatic as well as the tumorigenic properties in therapy-resistant cancer. Again, I've shown some more lab data on clonogenic activity, migration, invasion of these cells. So in vitro, it does work very well in these assays, including the sphere formation. Then we also did in vivo assays where we took enzalutamide-resistant prostate cancer cells alone or with the enzalutamide alone or with enzalutamide and this drug combination, as Dr. Talwar told you. In patients, you cannot just give a new drug. You have to also give the standard of therapy, which is enzalutamide, in prostate cancer patients, and we can see clearly that you can reduce these tumor growth from the resistant disease just by targeting this pathway. And some more data on the deep sequence data where you could look at multiple genes that could be affected from this pathway and the sphere formation, not only in prostate cancer now, therapy-resistant prostate cancer, but also some data we show you here for the pancreatic cancer cell lines, where you can have impact on the tumor formation in these models. So, irrespective of it, whether there is a normal cap-dependent translation process or a reprogramming process, we can use EI4G1 as a potential target for therapeutic intervention. So, I mean, to just go back to the history of the world, what we call antibiotics are actually antibacterial agents which only target protein synthesis. Erythromycin, tetracycline, chloramphenicol, and aminoglycosides, they are protein synthesis inhibitors for bacteria. Why? Because they selectively block the 70S bacterial ribosome and do not potentially have a less ability to affect our eukaryotic ribosomes. Then there's cyclohexamide. I was remembering from Bharat's earlier conversation, he said lymphotoxin only was toxic when you were using cyclohexamide to inhibit protein synthesis. But cyclohexamide itself is toxic to the cells. It's highly toxic and cyclohexamide cannot be used. There is still an unmet need at this point for the agents that can specifically use targeting components which are driving translational reprogramming in cancer. So make this long story short, normal cells have oncogenic addiction and results in a local tumor formation because cells proliferate and they survive. Then they acquire additional changes and become these therapy-resistant tumors. And what is at the base of all this oncogene addiction is oncogenes can drive translational reprogramming and they depend on this translational reprogramming to do their functions. And how do they do it? This translational reprogramming selectively primes for the proteins. It selectively loads messages that are required for proliferation for the local control of the cell growth, but also selective loading of the messages that are required for cell survival, migration and invasion are critical for driving the tumor progression from an indolent disease to an aggressive disease. And what we believe is that the EGFR41 is a critical component of this translational reprogramming process. As such, using novel agents, and nothing better than this conference to bring it, the small molecules or nutraceuticals that may target EGFR41 could have a huge impact on our therapy-resistant tumors in future. With that, I would like to acknowledge my contributors and thank everybody who was part of this discussion.
Thank you very much.
Thank you very much, sir. The session is open for questions.
Dr. Singh, can I make a comment and ask a question, please?
Yes, please.
Yeah. Thank you so much, Dr. Kaul, for showing us this wonderful protein that you talked about. This is a eukaryotic translational initiation factor 4, EIF4G as you call it. What is more important, and to our point of view, that this protein is increased significantly in patients of prostate cancer who have a very bad history. And in fact, the survival, if this protein is very high, is minimized. And you also showed us that one can knock down this one or disrupt the EIF4G, the EIF4E complex, and then it could actually be very, very helpful. I was just trying to understand, have you looked at this protein? Of course, you said that you looked into all the available cancer cell lines, but in addition to prostate cancer, any other solid cancer or solid tumor or in leukemias? What is the status of this protein in leukemias?
Thank you for that excellent question. We have only looked in the solid tumors, and almost every solid tumor that we looked at, we find that this protein is highly upregulated, or more than 200-fold levels compared to normal cells. Indeed, normal cells in many cases do not even have EIF4G1, but the tumor cells have high levels of this message, at least. Not the protein level, but the messages we have looked at.
So the prognostic value of this one that we showed in prostate cancer, is it also shown in other solid cancers?
Yes, sir. All solid tumors, the median survival of patients is at least 2 times as much when EIF4G1 levels are low.
Yeah, so I mean, breast cancer or maybe hepatic or
let me bring that data on screen so everybody can see that. This is the data for multiple tumors. Can you put the screen back on? Yes. 1 minute. You can look on the screen, he's going to share it with you. So if you look at this slide, these are the median survival of the patients based on how much of message for EIF4G1 is associated. I think the first one is brain cancer, gliomas, second one is kidney cancer, third one is liver cancer, fourth is lung, mesothelioma, pancreatic cancer, this is prostate, and this sarcoma. There were other tumors that is not listed because the list is long, but these are the data you can clinically look whether they are significant.
Fantastic. Thank you so much. That's really very, very good.
Thank you, sir.
Thank you, sir, for the nice presentation. What happens inhibiting this particular protein? What will happen to the normal cells?
Normal cells, nothing happens because this protein is not even present there. Very low levels are there. The cap-dependent translation is selectively used during the stress phase of the cells or during the repair of the DNA. When they have DNA damage, they require this process, or when they are in hypoxia. So most of those processes are critical for the tumors. In terms of normal stem cells, if you look at, we have not looked at it, but there may be something there. But in mice, there was no change in either histology or the weight of the mice during the treatments.
Thank you very much.
Thank you, sir. Go ahead.
Thanks for a wonderful talk. Thank you so much. We learned a lot from this.
Dr. Koul, thank you very much for a nice talk. In this mRNA, have you looked for the antisense mRNA also for this thing?
And can you repeat that?
Antisense mRNA.
For what?
For EIP factor?
No, we have not.
Thank you.
Okay, there is one more question.
Very illustrious answer. Thank you so much. Yeah, the question is, it was a very nice observation where in the normal cell this guy is not there. Do you have any idea the knockout, whether it is viable, the animal, the knockout, because certain critical guys, they may not be, but in development they have a lot of roles. So do you have any idea?
There are 2 knockout mice with EIF4G1. They are pretty viable, and they have smaller body weight, but other than that, they are living.
Oh.
It is not required for development.
That's very interesting. So, a quick question. Is it only one gene for this guy, or there are any?
Only one gene.
Only one gene.
But there are 3 isoforms. I told you. EIF4G has family of 3 members.
Yeah.
1, 2, and 3.
Yeah.
1 is selectively upregulated in tumors. 2 is for non-cap dependent translation in every cell, And the three wonders, gap-dependent translation, is in some cells. So one is selective isoform that we target.
Thank you so much.
Thank you very much to Paul
Thank you.
for illustrious, very nice lecture.
Thank you.
On metabolic, people were talking of metabolic reprogramming in cancer. And translational reprogramming is coming up as one of the next targeted therapies. It was really inspiring. Thank you very much.
So, I mean, these are not, many times when we sit in our own labs and in our own rooms, we just think only what we do,
Yeah.
but they are all interrelated.
Yes.
Any process in the cell is driven by proteins.
Yes.
Metabolism is driven by enzymes. And if you have enzymes in a different shape and form, we get different things.
And
But
metabolic reprogramming is not possible without
Translational.
reprogramming. Thank you very much.